their eyes are very small, but the cells that make up the retina are relatively large compared to most other species. So that makes for a happy preparation to go ahead and use. Now, I don't know if you guys know, but you guys are diploid organisms. And it turns out that these guys are tetraploid. And it's thought that that may be one reason why they are, they have these large cells. They've got all of that extra DNA, but it's not clear exactly why. So at any rate, we use these. And to do our experiments, we have to dispatch the animals. So we anesthetize the animals in a anesthetic for about 10 minutes. Then what we're going to do is to make a cervical transection and pith the brain so that it doesn't feel any pain anymore. And then what we do is we take out the eye, we cut the eye in half, we strip off the retina, and we put the retina in a little test tube that has a salt solution that mimics its normal extracellular fluids and also has an enzyme called papain that loosens the adherences between different cells. And we let it soak in that papain for about a half an hour, and then we wash out the papain, mechanically agitate the retina up and down through a little Pasteur pipette, and the cells come apart. And so what we're left with are single isolated cells. So that's kind of cool. So now I'll just go back here. And I should say, a lot of this research gets done over there in my lab. This is the view from the veranda. I wish it was my lab window, but it's not. I have no windows in my lab, so I have to go out to the veranda to get a breath of fresh air every now and then. But during the summer months, I've been lucky to do research at the Marine Biological Labs out in Woods Hole, Massachusetts. This is on Cape Cod, and it's a beautiful area. And they have a variety of fancy microscopes and other equipment that make it attractive to do research there. The other thing that they have are a unique set of animals that one can work with. In addition to tiger salamander, we also do work on an animal called a skate. It's like a stingray without a sting, and that's really kind of cool. Again, certain of its retinal cells are really large, and it makes it easy to do certain experiments. So that's why we would do that. If you want to get involved in research, you should know that there are research experience for undergraduate programs here and around the country for the summer months that you might want to get involved with. And that can be a lot of fun. I used to run one of those programs here for 6 years. I was co-director of the Marine Biological Labs REU, or Research Experience for Undergraduates program. Actually, I should just show you, because many of you are freshmen here. So I just want to show you very briefly. If you go to Google and you type in NSF, because that stands for National Science Foundation, and then REU, that's for Research Experience for Undergraduates, here's what you find. You go down to where it says Biological Sciences, and what you will find is a large number of programs at various places in the country, Boston, CUNY, Iowa, a bunch of different places around the country. And all of these different REU programs have different foci. So for example, here's one on plant genome research. Probably not what you guys are going to be interested in, but still, there might be something here that is something interesting. Like, for example, ecosystems, some of you might be interested in, maybe not another plant biology. Here's one, nature's machinery through the prism of physics, biology, chemistry, and engineering. Here's one on marine organisms in environmental change. and all different types. So you might want to look into these as possibilities to do summer research. One of the cool things about most of these programs, actually everyone that's labeled here, they pay you to do the research. So they pay you $5,000 for a stipend for the summer for 10 weeks. And they usually pay your room and board as well. And some programs will even pay to fly you in and out. So again, all of these different programs have a different emphasis. So here's one on, ecological data monitoring. Here's one on evolution, ecology. Gosh, there's a lot. So you're going to want to look through, and this is only one page. There's 5 pages of these programs. Just go ahead and show you here. Here's the second page. And if you take down the second page and you scroll down through it, you're going to find the Marine Biological Laboratory one, and I'm still listed as being the co-director. So, if you were to go there, you'd go to their site, and you get a lot more information, like here with the details here. Here's a fact of all of these programs. Students at the end of these programs give 15-minute scientific presentations. They write an abstract of that, and so these are all of their abstracts, and these are the videos that we actually had them broadcast over the internet. So that was really cool. And the students gave stellar presentations. I mean, really good presentations in my opinion. So you can go and click on this and take a look at what those presentations were like and get a feeling for what their kind of stuff was. Let's see if we can do that. So here's one. Okay. Past and future of bioluminescence.
Yeah, it's right. Okay. Hi, everyone. Sebastian, I'll take Sebastian's place as well here at the moment, and just remind you to go ahead and turn off or silence your cell phone.
And also to do copy editing for the English inside of it.
So this is
so I try to just
one student's presentation talking about
this is
cool work.
One of the ones that we worked on to clone the fluorescent proteins. This is the white light view and the blue light view again. This one has 7 different genes for different colors of fluorescent proteins.
So, really cool stuff they're doing with a variety of different organisms. You can take a look at it and you can get a feeling for whether you might be interested in trying your hand at this kind of stuff. So, a lot of programs, a lot of support from the NSF, a lot of opportunity for the summer. So, just something to think about. Okay, so, yeah. So, most of my work focuses in on what goes on in the retina. I use the retina as a model system to understand how nerve cells throughout the brain operate. This is a schematic of the different cell types, and I would argue that this little sheet of neural tissue uses the same molecular mechanisms to process neural information that is used throughout the brain. So if I understand the mechanisms by which these cells operate, I'll have a good general handle on what's going on elsewhere throughout the brain. And another reason to choose the retina is that it's easy to get at. It's easy to get isolated cells and retinal tissue. It's easy to stimulate appropriately. I can just put on lights and these photoreceptors will respond appropriately, and you'll watch the signals as they change from one cell to the next. And so that's why I choose the retina. I happen to, as a graduate student, got my PhD in doing retina, and I like the thought that I could address almost any kind of neuroscience-related question in terms of cellular and molecular mechanisms using that preparation. So most of my work has focused in on that. And you have photoreceptors that catch the light. They release neurotransmitter that's going to result in electrical changes in these postsynaptic cells. These ganglion cells are the output neurons of the eye. And here's one of those glial cells that I mentioned. It's marked M because it's also called a Mueller cell, after the fellow who discovered it about 100 years ago. And you'll notice that this guy has processes that cause it to radiate out between the synaptic connections of the neurons. So it wraps and envelops virtually every synaptic cavity. And that puts it in a really sweet position to be able to modulate neuronal activity. It's in the right space. And again, we think we have a cool mechanism by which it does this. And that cool mechanism relates to a really potent modulator of neuronal activity. As far as I know, this is the most powerful, naturally occurring modulator of neuronal activity that's in your body. And I'm going to show you an example. What we're going to do is we're going to take an electrode, we're going to stick it in a horizontal cell here, and we're going to see what happens to the response of that horizontal cell to flashes of light that the photoreceptor is sending to it when we change the concentration of this highly potent substance. So let's do that. There's our sheet of retina. I'm going to stick it with an electrode. And when we turn on the light, with a relatively low concentration of this inhibitory substance present, in fact, it's about 15 nanomolar. This is the response from the horizontal to flashes of light. There's a big voltage change here. It's about 30 millivolts, and that's a big response. And again, that's with 15 nanomoles of this potent substance. If I then add a little bit of more of that potent substance, it's going to be about 30 nanomolar now. Look what happens. So the cell first hyperpolarizes deeply. I mean, as much as the light was inducing, right? And the light-induced response to the same intensity flash has dropped by about 2/3. Now, if I increase that concentration of that highly potent substance from 30 nanomolar to 60 nanomolar, you see the following. Here, the signals are virtually abolished. In other words, synaptic transmission has been completely silenced. That's a potent molecule. It's reversible. If we go ahead and go back to the 30 nanomolar, we get a big increase in the response to light, and it depolarizes. And if we go back to where we started, the response is also good. So what is this really potent substance? It turns out to be nothing other than acid, pH. Just changing the pH of the extracellular solution from a pH of 7.8 to 7.5 to 7.2. By 7.2, there is no more synaptic transmission. That's one of the reasons why it's so critical that your nervous system maintain a stable level of pH, because otherwise, at this pH, your pH is about 7.38 in your blood. If it drops to 7.2, you're close to death, as it turns out. So pH is really, really, really a potent modulator of neuronal transmission. Okay. Whoops. So now I'm going to show you where the glial cells come in. And that's going to be over here. So first I'm going to show you this picture here. This is a beautiful drawing by Ramon y Cajal, one of the fathers of neuroscience, in about 1890, 1900. This is a drawing that he did of the cell types in a retina. I don't remember what type of retina this is. It's probably a mammalian retina. Photoreceptors up there. Second order neurons down here. They make synaptic contacts here. These are the ganglion cells, the output neurons of the eye. And this is one of those glial cells. And what I want you to appreciate here is this fine set of hairs, these fine fibers. These are the guys that are going to wrap and envelop virtually every synapse. And they also happen up here where, again, we get synaptic connections from the photoreceptors to the second order cells. So again, these glial cells are in a sweet area to be able to modulate neuronal activity.